Hello, and welcome to this Nature Research Custom Media webcast titled Going Out of Phase, A Tale of Protein Aggregation and Neurodegeneration, RNA to Explore Early Events in Protein Phase Separation and Phase Transition. My name is Nikki Forrester, and I will be your moderator. Today's webcast is produced by GenScript. We'll begin the webcast with a presentation from Dr. Elsa Zacco of the Italian Institute of Technology. We'll end the webcast with a Q&A. To ask a question, just type it in where it says ask a question and then press submit at any point during the webcast. We'll do our best to answer all of your questions. And now over to Dr. Zacco.
Hello, welcome everyone. I am Elsa Zacco and I am very happy to be here today to talk a little bit about the role of protein aggregation in neurodegeneration and how RNA is involved in this process. So, the cell has an important role in orchestrating and regulating the action and the interactions between DNA, protein, and RNA in order to coordinate the gene expression and regulation. This is often achieved by organizing both proteins and nucleic acid within macromolecular assemblies. These assemblies, they allows the protein and nucleic acid to come in close proximity with a higher concentration, can form condensates of both protein and nucleic acid, and sometimes they have a specialized function. When this occurs, these new structures are called membraneless organelles. You see some examples here. These organelles are characterized by the absence, of course, of a membrane, and they form by a process called liquid-liquid phase separation, in which the flexible proteins of the RNA, the constituents of the organelle, exceed the solubility limit and condense into liquid-like droplets. The biochemical properties of the proteins and of the RNA and the DNA found inside these droplets are different from the one outside. And therefore, the energy landscape, the thermodynamic parameter, and the interactions that they form need to change for maintaining the form of a liquid droplet within a liquid environment. The formation of membraneless organelles and the phenomenon of liquid-liquid phase separation is essential for the cells. This guarantees, for example, specialized compartmentalization, meaning that the intrinsic fluidity of the membraneless organelles allows the molecular diffusion while guarantees specialized recruitment of certain molecules and internal rearrangement. It's also important because the rate of in vivo reaction can be accelerated with incremental concentration within the droplets, and therefore the liquid-liquid phase separation could be a biological mechanism by which the cell tunes its reaction. Not only this, but also by trapping substrate of one cascade and excluding some others, the liquid-liquid phase separation can determine the activation of specific pathway of response compared to others. And therefore, it might be a means by which the cell regulates its own fitness. Among various membraneless organelles, I'd like to draw your attention towards the one called stress granules, which are cytoplasmatic MLOs or form of ribonucleoproteins, so proteins and nucleic acid complexes, whose function is to confer the ability to the cell to adapt to changes by tuning this biochemistry in response to intra or extracellular cues. The way in which stress granules do this is by promoting cell survival while condensing translationally stalled mRNA, ribosomal components, a translation initiation factor, and a group of protein that is called RNA binding proteins, which is very important from what I'm going to explain you later on. So what are the characteristics of the protein that find themselves within the stress granules or also other membraneless organelles? They often share an intrinsically disordered region, which is a region of the protein that of low complexity that favors protein-proteins interaction, bringing more proteins together in the close proximity. This, of course, favors the formation of higher local concentration and therefore liquid-liquid phase separation. Often this protein also have in common a prion-like domain, which instead, when interact with other prion-like domain, can help organize the protein into much more supramolecular structure, and the liquid-like droplets can sometimes develop to form a fibril-like condensate, thanks especially to the interaction between prion-like domains. Also, the presence of RNA binding domains within the protein is one important factor in the formation of these droplets. If we think, for example, of the certain protein have many RNA binding domains next to each other, we can imagine how much RNA they can scaffold upon themselves and how this can create a higher local concentration of RNA. It is also important to mention the role of post-translational modifications that can alter the protein surface charges and therefore strengthening the interaction between proteins, but also with nucleic acids. Of course, also the RNA that is inside the stress granules and other membrane-less organelles has an incredible important role in the formation of these condensates. RNA can in fact increase the local concentration of the RNA binding protein, acting as a scaffold for the interaction, but not only the length of the RNA, the simple presence of RNA, but also the type of RNA. In fact, let's imagine that more structured RNA can attract a certain type of protein, while single-stranded or less structured RNA favor the interaction with another set of proteins. This means that the RNA can tune the condensate biophysical properties and the composition and the viscosity of such membrane-less organelles. So under physiological condition, membrane-less organelles can disperse after their function has been fulfilled. However, there are certain conditions, and these occur frequently in many neurodegenerative diseases, in which one of the proteins that is found in cell, especially RNA binding protein within the stress granules or other membrane-less organelles, harbors a mutation that might render it more aggregation prone, so more prone to lose its native conformation and to aggregate. When this occurs, these proteins can undergo to liquid to solid phase transition, in which those liquid droplets become more static in time and can no longer disperse. The changes in the phase dynamic contribute to the formation of sometimes pathological persistent stress granules, and these are frequently hallmark of neurodegenerative diseases. We have here some examples. The examples in particular of frontotemporal lobar degeneration, amyotrophic lateral sclerosis, Parkinson's disease, and Alzheimer's disease. In each of these conditions, we found one or more than one RNA binding proteins found aggregated, or so they have transited to a more solid-like state. And you can see here on the top some pathological aggregates in tissue, and you can see how we can visualize them in cell. So unveiling the molecular mechanism of the RNA binding protein interplay may help us preventing aberrant phase transition. So within the cell, the interaction between RNA and RNA binding protein is sometimes essential to determine the fate of the cell. The RNA binding protein can control the localization and the usage of RNA, in part also through the formation of these stress granules. What happened, as I mentioned before, is that sometimes the genes encoding for RNA binding proteins involved in stress granules formation and stress response often harbor this mutation that are associated with increased propensity to aggregate, characteristic of neurodegenerative diseases. So what happened is that if upon stress, the normal course is the formation of stress granules in which the cell recruits many RNA binding protein in RNA and translation initiation factors, and this can get easily dispersed when the stress is being removed. In case we have this sort of mutation, and in the cases in which the protein, the harbor this mutation became more structurally unstable, the stress granule can become permanent and toxic. We have just a very short video here, in which you can see on the left, cells who've been just briefly treated with a certain stress in the formation of stress granules. On the right, instead, the stress is constant, and therefore the cells is not capable to disperse the stress granules anymore, and they become permanent and much more solid. I would like to talk to you today in particular of the case of a certain protein in the diseases of amyotrophic lateral sclerosis and frontotemporal dementia. Amyotrophic lateral sclerosis, ALS, is also known as motor neuron disease, while frontotemporal dementia is a clinical representation of frontotemporal lobar degeneration. Despite they interest different portions of the brain, they are now considered as part of one broad disorder, since they're linked clinically, pathologically, and mechanistically. And the breakthrough linking the disease mechanism for ALS and FTD was the identification of a common protein inclusion formed by certain RNA binding protein. Among these proteins, the protein called TDP43 was found to be the major RNA binding protein found aggregated in both sporadic ALS patients and some pathological form of FTD. TDP43 stands for transactive response DNA binding protein 43, and is formed by N terminus, which contains also nuclear localization signal, as two tandem RNA recognition motifs, and a disorder C terminus, which holds the majority of mutation associated to neurodegeneration. It is a protein that is mostly nuclear, where it binds both DNA and RNA, and has an essential role in transcription and translation. It is also involved in mRNA splicing, degradation, and stabilization. And it prefers to bind, it is important for something that we're gonna see a bit later, it prefers binding single-stranded UG-rich motifs. And it's been determined that is a protein essential from the neural development until the adult neuromuscular maintenance. So what happens in this disease is that TDP43 mislocalizes from the nucleus to the cytosol, where it's found aggregated, and this phenomenon is strictly linked to neurodegeneration. It has also been proposed by many that this aggregation passes through the formation of these liquid droplets that then in time become more static and solid, and they allow for the formation of these pathological aggregates. So ALS and FTD are diseases with a gradual onset, and their initial symptom can be quite varied between different peoples, and diagnosis is hard. It often occurs at a quite advanced stage of the disease. The lack of an accurate detection of the very earliest stages of protein phase transition has slowed down the understanding of the key steps of the disease mechanism, and therefore has limited the possibility to intervene on time. Today, I'm going to explain to you how we think that RNA that can be designed selectively to bind a specific aggregation-prone RNA binding protein may represent a promising therapeutic means to prevent aberrant protein phase transition, and also a concrete diagnostic tool for the early detection of disease molecular phenotypes. So we're going to talk about how RNA can stabilize the native conformation of RNA binding proteins, and also how we can use and exploit RNA for tracking protein phase transition. So we have established until now that the interactions between proteins and RNA play an active role in many neurodegenerative diseases. It would be enough to think, for example, of the consequence of the sequestration of many RNA binding protein in these permanent membraneless organelles, and the consequent aberrant RNA processing that would be caused by this. It has been reported in literature that RNA can have many different roles when it comes to protein. Among these, there is, for example, the proposal the RNA can be used as a solubilizer. This is an example in which we have the liquid-liquid phase separation of different proteins involved in neurodegeneration, and how this can be disrupted by the addition of RNA. On the other hand, it has also been proposed that RNA can act as a chaperone by suppressing the aggregation of a certain low chaperone substrate. So, if it is true that the RNA might modulate the stability of the proteome, how does the binding of RNA influence the structure of the RNA binding protein? And can protein aggregation be blocked through the interaction with the RNA, which is the natural binding partners of RNA binding proteins? So we try to answer this question by taking the case of TDP-43. And we started studying how TDP-43 aggregates, first of all, in vitro. I show you here simply a theoretical aggregation curve, which is a curve in which we use a dye intercalator of protein aggregates, so we can follow by the changes in the fluorescence intensity in time the evolution of certain aggregates in vitro. So we have a lag phase in which we have no fluorescence, meaning the protein is still in its soluble form. There is an exponential growth in which the fiber and the aggregates start forming until we reach a plateau phase. The case of TDP-43 in vitro is quite peculiar because it starts aggregating immediately, and it reaches, in our condition, a plateau after 24 hours. When we look at the aggregates formed in vitro by TDP-43 by itself, we can see that they look like this mesh of fibrillar mesh, sort of cotton-like, which still have some reminiscence of some fibrillar structures. So what we wanted to do is, let's take TDP-43 and let's see what happened when we add a different type of RNA. Let's see whether RNA can indeed be used to control the aggregation process, or the phase transition process of TDP-43. So as a reference here, you have again TDP-43 in the absence of any RNA and how it looks under the microscope. First of all, if you remember, I mentioned before how TDP-43 binds preferentially single-stranded UG-rich RNA. So what we've done here, we have taken a very short stretch of RNA, around 12 nucleotides, and rich in UG, and we try to incubate it with the protein to verify what happens to the aggregation of TDP-43. As you can see, with different amount of UG-rich RNA, the aggregation of TDP-43 in vitro was substantially reduced. And the more RNA we gave, the more the effect seemed to be strong. As a control, we took also the reverse complementary sequence of this UG-rich RNA, so let's call it a CA-rich RNA. And we see that in this case, we had instead no effect on the aggregation rate, so on the speed in which the protein aggregates in vitro, nor on the morphology of the aggregates. We wanted to determine whether this was a phenomenon strictly sequence specific or if it depended by something else. So we have created two scrambled versions of this GU-rich RNA, and we again incubated with the TDP-43 and follow the aggregation of the protein over time. So we could see that with increasing concentration of these two scrambled version of GU-rich RNA, we do have indeed a significant reduction of the aggregation of TDP-43. However, not as strong as it was in the original case. So sequence, it is definitely important. When we did the same with the controller CA-rich RNA, again, we had no effect whatsoever. As a last point, we also wanted to verify the role of multivalency in the phase transition of TDP-43. So we have taken our initial GU-rich RNA and we created a longer version by simply repeating the same sequence 3 times. What we noticed is that we had a significant drop in the aggregation of TDP-43 in vitro until we completely abolish the phase transition of the protein already from the lowest molar ratio protein RNA. And not only we could barely identify any aggregates, but the ones that we did identify, you can see, you can appreciate, I'm sure they look very different from what we observed before, and they seem to be at very earlier stages of the aggregation process. Almost like if we are still in a transition phase between the liquid phase transition and liquid to solid phase transition. Also interesting, when we recreated the same longer RNA version with our CA-rich RNA, we observe actually increment in the speed of aggregation of TDP-43 and also a much denser aggregates, which almost became completely amorphous. So the next step is, of course, how we can use these to try to prevent the aggregation of TDP-43, at least in cells. So here you can see some cells which have from the molecular point of view, phenotypically associated, or it can be related to ALS, meaning that some of these cells, TDP43, that is normally nuclear, you see here in green, instead starts to mislocalize from the nucleus to the cytosol and start forming these little cytosolic aggregates. So what we did in this case, we have incubated again our cells. We have given to the cells our GU-rich RNA or the CA-rich RNA. In the case in which we gave to the cell the GU-rich RNA, we could observe that the amount of soluble TDP43, so not aggregated TDP43, was incremented in the presence of GU-rich RNA and reduced instead with the opposite sequence. And also the number of the cells with aggregates has been is directly proportional to this. Another important point is that we observe by fluorescence recovery after photobleaching, and these are quite preliminary results, if the aggregates formed by TDP43 by itself are almost completely immobile. Instead, when we add our UG-rich RNA, there is an important recovery of the dynamicity of this aggregate, meaning that the aggregate form in the presence of this RNA are much more flexible, and therefore there is a higher chance for their dispersal. So if the RNA modulates the behavior of TDP43, this means the RNA might be designed to prevent the aggregation of a target protein. So I would like to continue by also telling you how we can use RNA for tracking the protein phase transition. And in this case, I have to introduce a new concept with the RNA aptamers. Aptamers are strong-binding single-stranded oligonucleotides that target a biological molecule of interest. And they can bind the proteins with affinities derived of those of monoclonal antibodies. Some of them have already been implemented and have many potential uses in intracellular processes studies, but also in medicine and technology. The main advantage of using RNA aptamers over antibodies is, first of all, their size. Being much, much smaller, they show a higher binding density due to fewer steric restriction, but also their chemistry, which in fact, small oligonucleotides are easier to synthesize and modify and suitable for large-scale synthesis with a higher efficiency and lower costs. There are, however, some limitations. First of all, we are talking about oligonucleotides, therefore they can be subject to nuclease degradation and structural instability, which, however, can be improved by introducing certain chemical modification. But most of all, until now, there was the problem of their complex generation, which was until now achieved solely by the sequential evolution of ligands by exponential enrichment, a protocol called SELEX, which via a series of a cycle of improvement try to optimize the sequences starting by a random pool of oligonucleotide that bind a certain protein. I would like to propose to you today instead an alternative to this, which is the in silico design of RNA aptamer for, in this case, a specific case for TDP43. What we've done here is that we have used an algorithm called 𝗓𝗏𝗏, which estimates the protein-RNA pairs binding propensity by combining secondary structure, hydrogen bonding, and van der Waals contributions. It is possible to start either from a pool of known RNA interactors or starting from the whole transcriptome. And what the algorithm does is dividing the RNA binders into smaller fragments and predicting the individual binding of these fragments to each portion of the target protein. Once the software identifies the predicted best RNA binders for a given protein, it compares it with a reference set to determine also the most specific aptamer for the target. So what we've done is exactly this. We wanted to design the novel RNA aptamers for TDP43 and to evaluate whether we can use it to track TDP43 aberrant phase transition at the initial stages. So we started with our algorithm 𝗓𝗏𝗏, and we have taken the whole list of known RNA binders of the protein. We had the clip data available, so this was easily doable. And we generated a list of fragments along 10 nucleotides, and we moved over a slightly window of 10, one nucleotide at a time from the 5' to the 3'. We had therefore this very, very long list. And for each of these sequences, we calculated what we call the RNA fitness score. This score is derived from the prediction of the interaction propensity of each of these fragments with our target protein, and also is a consequence of bombarding these fragments with a series of mutations to try to improve upon this predicted binding. So once we have our pool of best binders, best predicted binders, we also calculated and determined the protein fitness score, meaning that we have measured the strength of the interaction propensity of each of these fragments in comparison to a pool of protein of the same length and the same amino acid composition of our target protein. In this way, we generate a series of short RNA aptamers that are not only designed for our target protein, but they are mostly specific for our target protein. So the next step was, of course, validate whether what was predicted by the algorithm 𝗧𝗶𝗺 was indeed correct. So we had to determine the interaction and the binding strength between the RNA aptamers we designed in silico and TDP43. We started by doing this in vitro, so we produced TDP43 in E. coli and determined the binding affinity with the aptamers. And the way in which we mostly do it is by using a technique called biolayer interferometry. It is a technique based on the interaction of light waves, in which the white light is sent to the certain glass fibers and reflected back. And when the binding occurs, the path length of this reflection increases. But the more molecule bind, the more the interferometric profile shifts to the right. Therefore, when we plot the magnitude of the shift as a function of time, we have the classic association-dissociation curve. In our specific case, we had some streptavidin biosensors, and we biotinylated our RNA aptamers. We loaded them on the biosensor, and after blocking, we added our protein at different concentration to determine the shift and therefore to obtain these association-dissociation curves. So I show you here the examples of a few aptamers that we tested. We determined by this bilayer interferometry technique the KD of binding, so the dissociation constant. The lower the KD, the stronger the binding. You can see here that we find some very, very good candidates. I show you here the binding response curve of one of our best aptamers, which has a KD of 16 nanomolar, and one of the negative control, in which instead the KD goes to the micromolar range. So not only we were happy to see that what we designed in silico was indeed binding to the TDP43, but we also noticed a very important correlation between the experimentally determined KD and the 𝗧𝗶𝗺 predicted interactions. So now that we have our favored RNA aptamers designed specifically for TDP43, we wanted to evaluate whether we can indeed use them to track the aggregation of the protein from the early stages. So we used the RNA aptamer as a probe in experiment of a PAINT super resolution microscopy. So PAINT stands for Points Accumulation for Imaging in Nanoscale Topography. And what we did is we conjugated a fluorophore to our RNA aptamer, and we allow it to bind and associate and dissociate with our target, taking many pictures at the same time. This creates a sort of a blinking map, which allows us then to recreate the image at high resolution. So what we have here is the comparison between the images created with using our RNA aptamer for TDP43 in time, and also images derived instead from the most standard state-of-the-art molecule used for determining the presence of protein aggregates, which is thioflavin T. So as you can see, what to me is the most important message is that thanks to our RNA aptamer, in conjugation with super resolution microscopy, we were able to identify the smallest oligomers of TDP43 as small as 5 nanometers. and also already at the very, very early stages at the time zero of our experiment. And these are aggregates that with standard technique cannot be detected at all. So our hope is, of course, that this level of tracking such smaller species can be then applicable to for potential diagnosis. So to go from this on, we have tested, first of all, if we could reproduce the same results in cells. And so we have, again, here certain cells that have expressed TDP43 in a condition in which the protein is induced to aggregate. So again, the TDP43 is in green, and we have our aptamer in red. What we see here that we obtain a mixed population of cells that have TDP43 in their physiological nuclear localization. In some cells in which TDP43 has shifted from the nucleus to the cytosol, and other cells in which they clearly present aggregates of TDP43. So no matter in which state the TDP43 was found, either soluble or mislocalized and aggregated, wherever the green of TDP43 was, we found the red of our aptamer. And this is very important because you can see clearly here from the fluorescence profile that the green and the red go always hand in hand. But the essentiality of this is that this means that it will probably, the use of this aptamer for binding selectively TDP43 should be able to allow us to track all stages of TDP43 in all its form, starting from the soluble towards the solid-like stage. This is just to show you, as we did also before in vitro, that these RNA aptamer are specific also regarding the sequence. When we use a control RNA, we do not observe at all the co-localization with the TDP43 that we have instead observed when using our in silico design aptamer. We move then to tissue, and these are preliminary results. We are trying to verify whether it's possible to use, of course, our RNA probe to visualize the small aggregate of TDP43, in this case, in post-mortem ALH patients' cortical section. So we have stained the section with anti-TDP antibodies, anti-aggregated TDP43 antibodies here in green. And then again in red, you can see the same done with our RNA aptamer. So first of all, it's clearly to say that we can obtain a much higher resolution and specificity using our RNA aptamer compared to the standard used antibodies against TDP43 aggregates. And this allowed us to determine the aggregates number and length in comparison to an age match control. Last but not least, I would like just to show you briefly what we are heading towards from now on. So as I say, the main scope of this would be to use these RNA aptamers to be able to track at the earliest stages the misbehavior of TDP43. So the formation of the oligomers TDP43 in living patients. So we are starting by studying whether this is possible to do in cerebrospinal fluids of ALS patients. You have here one case in which we have compared the efficacy of using our RNA aptamer to target small TDP43 aggregates against any other RNA with other sequences. These differences is important and is significant. So we believe the RNA aptamers offer an improved substitute to currently used detection methods for phase separation and phase transition, and might help to unveil the mechanism behind aberrant aggregation, allowing therefore for early diagnosis. Just to sum up what we talked about today, aberrant condensation of RNA binding protein involved in stress granule formation, it is often linked to neurodegeneration. The formation of these permanent stress granules entrap indefinitely both RNA binding protein and certain mRNAs and can lead to toxicity and cell death. So understanding the interplay between RNA binding proteins and RNA in both physiology and pathology is essential to unveil the molecular mechanism behind the neurodegeneration. To do so, we need to be able to track and see the aggregation of the RNA binding protein at the earliest stages, and also to design new therapeutic tools that can stabilize both structurally and functionally aggregation-prone RNA binding proteins. So what I told you today is that maybe RNA aptamer could be the answer to both these needs. So we see how with using RNA aptamers, we can have reduced time and cost of production by designing them in vitro via 𝗓𝗏𝗾, in silico, with 𝗓𝗏𝗾. We can improve the specificity towards our target by predicting the potential unwanted interaction with the rest of the transcriptome and the proteome. We can design the RNA aptamer for tight and specific binding of TDP-43 involved in both ALS and FTD, but also other diseases such as Alzheimer's disease. And what we're doing now is testing these aptamers for their ability to block or slow down TDP-43 aberrant self-assembly. And the one instead that we have designed via 𝗓𝗏𝗾, they've been used to track the phase transition at the early stages, both in vitro, in cell, in tissue, and now hopefully in cerebrospinal fluid. So with this, I conclude, but please allow me just a few seconds to thank all the people that worked on this project, but main of all, Professor 𝗗𝗶𝗺𝗃, who is the mind behind much of this, and of course the funding. And I want to thank you all for being here. I would love for you to ask anything that you think you'd like to know, and we can have a nice chat about it. Thank you very much.
Thanks so much for the wonderful presentation, Dr. Zacco. So now it's time for our Q&A. If you'd like to ask a question, just type it in where it says ask a question and then press submit. All right, and I see we've already got a few in here. So someone was wondering, could you expand on what you mean by stress and what stressors are?
Okay, sure. I mean, the stress granules are formed under response of different sorts of stress. Normally, it's specifically osmotic stress, oxidative stress, and also thermal stress. But this is only, I have to say, just one example of memberless organelles that involved RNA-binding proteins and are associated with neurodegeneration. For some reason, they have found, because they are so rich of RNA-binding protein, they are particularly, they go hand in hand with the fate also of RNA. But the same can be applied, it can be said for many other memberless organelles. I hope that helps.
Yeah, definitely. Thank you. Someone else said, thank you for your presentation. They were wondering which construct of TDP-43 you used in your in vitro aggregation studies. Just the RRM12, which is sufficient for RNA binding, or the full-length version of the protein?
In the case that I show, we have actually something in between, because we have we wanted to test the binding, so we started by testing only taking the two RNA recognition motifs, but then we expanded, also included the N-terminus, and lately also the full length with a solubility tag. So just for whoever is not familiar with this world of protein in vitro and how difficult it is to produce them when they tend to aggregate. A trick is, of course, to try to produce the portion of the protein you are most interested in. As this person from the audience correctly pointed out, the RNA recognition motif is the one that recognizes the RNA. So if we want to study the interaction and determine the strength, it would be sufficient in vitro to probably compare just the interaction, determine the interaction with this portion. However, the region that is responsible mostly for the aggregation is also the C-terminal. So sometimes you need to look at the full length, and they can be very hard to produce and purify in the monomeric version. In those cases, sometimes it's useful to add some solubility tags, like the maltose binding domain or the SUMO domain, that are known to keep and maintain the protein longer in solution for the purpose of the assay.
Great, thank you. Another attendee was wondering, how would the synthetic RNA be administered?
This is a good point. So we are currently in the stage of investigating what will happen at in vivo, because I have to say it's not our expertise. So we are collaborating with people who instead know how to do this, because when we study in cell, as this person obviously know very well, the way of administration can be very different from the ones that are used in vivo, in vitro. We can only take so far examples of what we know about how other RNA are administered in vivo, especially microRNA, because we are talking currently about oligonucleotides. They are as short as 10 nucleotides. So we are talking of very tiny molecules, can be longer, but they're very small. So we can learn from them and try to understand the best way. However, within the consortium that we have, we are trying to develop also a correct delivery system that is might be probably a mixture of lipophilic particle and cationic particle to be able to deliver, first of all, to primary cell lines and then hopefully in vivo. But if this person has suggestions, please, they are more than welcome. We are learning all about delivery now, so you have my email. Please write.
Someone else asked, how efficiently can you get the aptamers into cells?
We've been quite lucky on that sense. Not many people have done the transfection of RNA into cells. What we do, we normally, by associating them with fluorophore, we can estimate using standard techniques such as cytoperimetry how much of our RNA aptamer actually enters the cells. And we can arrive up to 80% in extreme cases. But on average, I would say that around 50% of the population, between 50 and 60 of the cell population contain our fluorophore. So we presume also our RNA. We had to optimize the commercially used transfection reagents, let's say. We had to optimize a little bit the protocol to fit to our the smaller size of our RNA. But after a bit of tuning, we're quite satisfied at the level.
Great. How was the TDP-43 RNA interaction in plasma?
We will start it soon. we have to look at it soon. We haven't done it yet. And thank you for pointing out how important it is. It has to be done. Also because we need to, it's not incredibly abundant protein in the blood. So it's not as easy. That's why at the moment we're starting with cerebrospinal fluid. But in the future, it would be amazing if we could simplify it by looking into plasma.
Great. Can you visibly observe the protein phase separation?
So I did not show any live imagings. They're too preliminary, but we can actually see. We do see them with techniques such as microscopy techniques such as fluorescence correlation microscopy or with FRAP. These are techniques in which you grow your cells in your medium and you can go directly under the microscope while the cells are alive. And you can observe and study the physical chemical properties and the dynamics within these aggregates. So you can determine whether they are tough aggregate or they are small and maybe just liquid like condensates. And you can, in this way, study the effect of the RNA also on the properties of the aggregates they do form. I apologize, I know nothing yet permanent, definitely to show, but we are looking into that.
Another attendee said, great presentation. Is 𝗧𝗶𝗺𝗏 available and is it open source?
Yes, you can all go and play a little bit. So what 𝗧𝗶𝗺𝗏 it's currently, you can do a lot of things because it's a group of algorithms. You can find easily in the site, which I think it should be in the resources of this webcast. And you can easily predict the interaction between a protein of your choice and an RNA of your choice, but you can also do more than that. Do go and play with it because it's an amazing tool.
This might be a very similar question, but someone was wondering when you might expect your peer review to be completed.
So we have the data on on the tracking, let's say, the possibility of tracking TDP aggregates with the aptamer has been published just this summer in Nature Communications. And we consider that the beginning, of course, because there is much more work that we would like to do on this. In the side of the tracking, of course, we are working now with our collaborators on checking the cerebrospinal fluid, but also in the guts. And so it's going to be a big thing that will take a while, however. Regarding instead the usage of RNA aptamers for reducing or preventing the aggregation of TDP43, I hope soon. Well, it's not submitted yet. We are collecting the latest results, but I hope I'm not too positive to say next year.
Can RNA aptamer track how protein aggregates are being degraded? For example, via lysosome or autophagy?
This is very cool. I did not look at it in this way, but I don't see why not, really. I mean, we will have to have a system that allows us also to make sure that the reduction of the aggregate is not a direct effect of the aptamer, but is actually due to degradation. So I would say that we probably need more than two probes. But I will look into it. I don't think it's impossible. I think it might just be technically tricky, but doable. Thank you for the idea.
Can the same method of aggregate detection with RNA be applied to other proteins?
Yes, of course. We have chosen TDP43 because it's, as I explained in the presentation, it's the major RNA binding protein in these two diseases and in other. So it was some sort of was exemplary for our study. But we have prepared aptamers for other proteins. We are studying currently other neurodegenerative associated proteins in this way, but also proteins associated with other diseases. And so it's a field that is starting now, especially now that we have the opportunity to design the aptamer in silico, avoiding all the complication of the in vitro part. So I'm expecting to see much more coming soon.
Great. Can this approach be used to visualize and quantify small protein aggregates in living patients?
So the final aim is exactly that. I show you the very preliminary results. They are in cerebrospinal fluids of living patients. As someone else mentioned before, it would be amazing to have an even less invasive way of tracking this. However, it also depends on the actual abundance of the protein present in that specific fluid. But the final aim is exactly this, to be able to use such small, non-invasive and non-toxic molecules to determine and detect the smallest aggregates at the very, very early stages.
Right. Another attendee was wondering, how can the RNA probing be further improved?
So we are thinking a lot about this because for sure there is space for improvement, and something that we believe is definitely doable. It's adding certain chemical modification, has been shown also in by others who have used RNA as therapeutics. Let's say how of impact can be adding sometimes only a single modification on the RNA towards its function. So this same concept should be applicable also to our synthetic short RNA. And modifications, there are methylation, the addition of fluor, that can be done on the bases or on the sugar. Any position can improve the binding, but mostly can improve the stability. Because being such a short and small molecules, they might be easy to degrade. So the addition of certain modification might hide them from the nuclease, the cellular nuclease, and allow them to circulate for much longer.
How is this approach better than other ones that are currently available?
Well, I don't know whether it is necessarily better. We are trying to prove that it might be better than currently used antibodies to detect TDP aggregates, or in the future other protein aggregates. But it's definitely, it has the advantages of being easy to prepare. So these RNA are so small and so approachable in terms of costs that almost anyone could do it, and it is already, I think, a big step ahead. Also, because they're small, you can have much more RNA binding to the same protein. So this gives a signal of whatever you're trying to read, which is much stronger than other larger protein might, like antibody might do. And also, we are heard a lot talking about RNAs, especially now recently with the vaccine and so on. So there will be further improvement for sure, and the field, I think, it is going ahead pretty well.
Exciting times.
Yes.
All right. Well, I'm afraid that's all we have time for today. Thank you so much for your wonderful presentation and for answering everyone's questions. And a big thank you to our audience for being here with us today. We really appreciate you participating. I'd like to kindly invite you to answer a few brief survey questions at the end of this webcast, and remind you that you can always come back and watch this at any point in time on demand at nature.com webcasts. Thank you again, and I hope you'll join us soon.
Thank you. Thank you, everyone. Bye.